Welcome to the 6th episode of Sci-Five. I'm your host, Shea-Kurek. On Sci-Five, we talk about the most relevant technological and scientific innovations of this week. This week, we'll be talking about human gene editing, new advances in imaging technology, and a new discovery into how cyanobacteria see the world. A quick aside about the Advanced LIGO program that had a success this week in discovering gravitational waves. Congratulations! Hopefully this will advance science dramatically. This week, the UK Human Fertilization and Embryo Authority gave the first approval to use the CRISPR gene editing system permanently on human DNA. Researchers at the Francis Crick Institute, with the permission, now will be using the gene pair CRISPR and Cas9 in order to check out human embryonic development in the early cycles, which has not been studied. This is the first time that permission has ever been given to genetically modify humans to date. Scientists at the Research University of Rochester have created a new plastic that greatly improves the medical field. After being manipulated or stretched into a new shape, the polymer is capable of returning to its original form. Unlike other plastics, this memory-shaped plastic is able to reform with astoundingly low amounts of energy, that to the equivalent of the human touch in terms of temperature. Another unique feature of this plastic is its ability to store mechanical energy and release it when put under these types of circumstances. It's capable of lifting about 10,000 times its own weight. New imaging research is on the way this week at UCLA. As new technologies like smartphones and driverless cars become more and more common, computers' ability to process image become more and more relevant. The new algorithm they have developed, called the face stretch transformation, is a computational process to help computers see images and information that are typically not visible during standard imaging techniques. Throughout most of history, imaging has been done for the benefit of humans, not machines. This image development algorithm is specifically created with that in mind to help programs and artificial intelligence analyze the images and data from it more efficiently. UCLA has made the code for this project open source so that it can improve with greater efficiency. The interesting questions biologists have had for centuries about cyanobacteria reacting to light have been answered this week. Cyanobacteria is found in massive bodies of water around the world and is one of the most common and numerous forms of life. Their ability to see is very important for cyanobacteria as they use photosynthesis as their main source of energy. It has been known that they are acutely aware of their surroundings, but until this week it was unknown by what process they were understanding that. Scientists from Queen Mary University of London have discovered that in earlier studies of their cells that they create a lens, at which point they have photoreceptors and act as one of the most primitive and oldest versions of an eye. Although not as advanced as other eyesight, scientists have not known about these tiny slime balls' ability to see for the past 340 years since the invention of the microscope. The Department of Energy SLAC National Accelerator Laboratory has been working on a way to take atomic resolution images of biological molecules using low-quality crystals and X-rays. It has always been thought prior to these experiments that expensive high-quality crystals were needed to gather images of biological molecules. To their surprise, in the most recent discovery, biological machinery involving photosynthesis was being studied, and it was found that imperfect shards yield more precise results than previously expected. To date, well over 100,000 protein structures have been identified using Xray crystallography, and with this latest advancements, hundreds of thousands of previously unscannable molecules will be capable of being identified. That's all for this week. Thanks for joining me, and we'll see you next time on Sci-Five.